Hello and welcome to the second tutorial on using bioinformatics tools in further education genetics teaching. In this video we're going to look at how to use a tool called XBasy Translate, which is going to turn my nucleotide sequences into amino acid sequences. Before you start, you should at this point have 4 sequences from the first video. You should have the HOXA1 full sequence that we found together. You should have the HOXA1 sequence that only contains the exons, and it should start with ATG. After the video, you should have used your tools to go and find HOXB1 full, and then you should have also found the HOXB1 exon. Now, the HOXB1 exon was actually really difficult to find, and there wasn't a satisfactory one. So I have used one that starts with actually TGA. If you have used a sequence that is around 1400 base pairs in length and says complete CDS on it, then you're fine. If you're using one that says partial CDS, get rid of it, go back to NCBI and find yourself another one. So first off, what are we doing? So we're going to search for XBasy Translate. Been there before. And you're going to find yourself on a site that looks like this. So what this site does, as I said, is it's going to turn my DNA sequences into nucleotide sequences, and I have got a couple of ways that I can do that. So I'm going to demonstrate this using HOXA1, and I'm going to copy just the sequence, not the title with the chevron in. XBasy Translate doesn't like the chevron, but some other sites do. So we have to keep that in there for now. So I'm going to paste my whole sequence in there, and I've got 4 different output options. I have got something called verbose, compact, includes nucleotide sequence, and includes nucleotide sequences, no space. And I'm going to show you what all 4 of them look like. Sometimes it's just you want something very specific. Other times it's a case of that it's what looks better for you. So we're going to go for verbose first. So we're going to click translate. And what you have is it just has the single letter codes with stop codons. Now it looks a bit funny, like, don't know what is happening here. What is happening is that the program doesn't know where the start of the sequence is. So if we come over here for a second, let's get up our sequence here, and let's just actually make that. Okay, let's make this a bit bigger. So what you've got here is the sequence, but if you remember back to your codons, we know that the genetic code is only 3 letters in length, and it is non-overlapping, which means that each codon exists on its own. Which means that the first letter can either be the start of my first codon, the second letter can be the start of my first codon, or the third letter can be the start of my first codon. And we call this idea open reading frame. XBasy Translate has done that for me. So I'm going to come up here, and this time I'm going to click on includes nucleotide sequence, and we're going to compare these open reading frames together. So I'm going to click translate, come down, and here you go. So open reading frame 1 in the forward direction, so from 5' to 3', has got my first codon being ATC, which is isoleucine. If I scroll down to open reading frame 2, it's showing you now that my first codon is starting with TCA, which is serine. My third open reading frame, which starts with C80, which is histidine. It also gives me it in reverse. So if I was actually starting at the bottom of my sequence, so if I scroll down, all the way down to the bottom of my sequence here, it gets quite long. So it's translated it for me. So remember, A binds with T, C binds with G, and it's reversed it. We've called this, reverse complement. We don't really look at the reverse ones, and for all the ones today, we are actually not looking at them. So how do you know which one is correct? Simply, you look with the bit that's got the biggest bit of pink. So for us, that's this one. Open reading frame 2 really clearly has a nice big chunk of pink from my start codon here, methionine, all the way to my dash, my stop codon there. My other sequences look like so. So I can also have includes nucleotide sequence, no spaces, and it gives you it in its shortened form like so. It also has compact with no spaces, and then it looks like this. It gets rid of my nucleotide sequence for me. Now, in the previous video, we looked at my full sequence, and then we also got my exon sequence. So I'm going to do the same thing now with just my exon sequence to prove to you guys that in essence, it is only containing my actual nucleotides that I need. So I'm going to go for includes nucleotide sequence with no space. I'm going to translate, and there you go. Now, it starts at open reading frame 1 because according to this sequence, it starts with the first letter. So it's not the sequence itself, it's your letters that you have put in which will dictate whether it's open reading frame 1, 2, or 3. So what I'd like you to do with this video is pause, go back, follow exactly what I have done. Use translate to translate your full sequence. Check that you have got open reading frame 2 as well. And then you can also do your exon sequence. You can also do the same with your HOXB1 sequence as well, which open reading frame is correct. Thank you for listening, and I'll see you in the next video. And in the next video, we're actually going to look at aligning our sequences.